And now on to the last in our group of major organic macromolecules. We're going to be talking about the proteins, and then we'll also be talking about the nucleic acids. So proteins contain carbon, hydrogen, and oxygen, just like we saw in the carbohydrates and the lipids, but also will contain nitrogen. Sometimes there'll be some proteins that contain sulfur and also phosphorus. So just like before, proteins are polymers. The individual building blocks, or the individual monomers, are known as amino acids. There are 20 naturally occurring types of amino acids. There are some that have been experimentally created, but there's only 20 naturally occurring. They are joined together by a type of covalent bond called a peptide bond. So the type of bond between two amino acids is known as a peptide bond. They contain some really important functional groups. One type of functional group is known as an amine group. And if we look at an amine group will contain a nitrogen. So an amine group will contain a nitrogen and some hydrogens, hydrogen and hydrogen. Right. And it also contains an acid group, and that's our old friend, the carboxyl group. So it's our carbon double bonded to an oxygen and then a hydroxyl. Okay, so those are two different types of functional groups always found on our amino acid, and we'll see examples of how it's found on the amino acid later on. Now, amino acids sometimes can act like an acid and donate hydrogen ions to the solution, and sometimes can act as a base and remove hydrogen ions from a solution. All amino acids will have exactly the same structural formula, or exactly look very similar to each other, except for what's called an R side group. So let's have a look at some different types of amino acids. First, we have our generalized amino acid. All right, let's have a look at this generalized amino acid. All right, first I want to find the central carbon. So look for the thing that's R in green. That's the R side group, and follow the line directly down. That carbon, that's the central carbon. Everything is bonded to that carbon. If you look just over to one side, you'll see the amine group, the N, the nitrogen with two hydrogens. On the other side, our acid group, which is our carboxyl acid. Then we have just a hydrogen, and then up from that central carbon we have the R side group. Now compare that generalized structure to all these other examples of amino acids: glycine, aspartic acid, lysine, cysteine. We see that same structure. We have our central carbon. Attached to that central carbon we have three things that are identical in all of them: the amine group, the acid group, and the hydrogen. The only thing that is changing is that R side group. So in glycine, it's a very simple amino acid. The R side group is just another hydrogen. In the aspartic acid, we have a carboxyl group. So we say that it is acidic. It acts like it is acidic. Then we have lysine that has another nitrogen up there that has a tendency to pull hydrogen ions out of solution. And then cysteine, which is one of the amino acids that, or is the amino acid that contains sulfur. Now this cysteine is fairly important, and this sulfur is really important because sulfurs in very large proteins, two cysteines and the sulfurs will have a tendency to bond together. So that's going to affect the shape of the eventual protein. But let's right now just concentrate on putting two amino acids together. So here we have two generic looking amino acids. You can see in blue the portion that's going to interact with each other by a dehydration synthesis. So the dehydration synthesis will put these two together. You'll remove water and you'll create your dipeptide. That's what we call two amino acids bonded together. So they're bonded together by a peptide bond. You add the water back in by a hydrolysis reaction. and you break it apart into the two amino acids, the two monomers once again. All right, so next step, let's look at a whole bunch of these that are bonded together. All right, and we end up with something called the primary structure of a protein. The primary structure is just the sequence of amino acids. So this could be any sequence. Maybe, I don't know, we have a glycine bonded to a lysine bonded to, a cysteine. It's just whatever the sequence is. Now, all those different amino acids, however, have different properties. Some are acidic, some are basic, some have sulfur, some are very nonpolar and hate water, some are very polar and love water. So they all have different chemistries. So this primary sequence, what the sequence of these amino acids are, will eventually determine the shape of the final protein. And that's going to be a very important function, or a very important structure, sorry, not a function, a very important structure of the molecule that will determine its function. So the primary sequence, the first level of order of the protein structure, is really the most important. Then we have our secondary structure of any large protein. These are alpha helices. All right, so that term right there means alpha. And beta sheets. So that term right there means beta. All right, so we have an alpha helix and a beta sheet. These are just a couple of very typical structures that we see inside proteins. And then we have our tertiary structure. So this is where you have a lot of alpha helices or beta pleated sheets all folded up together, and it forms a very tight, compact molecule. All right? So it's one polypeptide. So a polypeptide is made up, that's a peptide, is made up of many amino acids, all bonded together by peptide bond. Right? So we have one polypeptide, highly folded, that's our tertiary structure. Now sometimes we'll have a quaternary structure. A quaternary structure is where you have two or more polypeptide chains, each highly folded, and then they fit together. Now, based on this final structure of our proteins, we can generally divide all of our proteins into two major groups. There's going to be a lot of subdivisions within this, but there are two major groups. First, we have our fibrous proteins. We often call these structural because they help form a lot of the structures inside the cell. They're fibrous, they're strand-like. They usually don't dissolve in water very well. They don't break apart, so they're very stable. They will have that tertiary or quaternary structure, that's their very complicated three-dimensional structure. Mechanical support, tensile strength, and that's maybe a new term for some of you guys. Tensile strength is the stretching strength. So that's tensile strength, is stretch strength. How far can you stretch a strand before it snaps? How much weight do you put on a strand before it snaps? So good examples of these include keratin, elastin, which as you can guess is elastic, collagen, the most abundant protein in the body, and some other contractile fibers. Now you may have noticed for some of these terms there's a little bit of IN. Whenever you see IN on something, it means it's a protein. Let me step back for a second. I forgot to mention for another one. Does anyone know what it means if you see OSE on the end of something? What was a What was a word where we saw OSE on the end? Well, glucose. So often if you see OSE on the end of a word, it means it's a carbohydrate. All right, so we had our fibrous proteins and now our globular proteins, functional proteins. That means that these are going to be, well, mainly enzymes. We're going to talk about them as enzymes. They're small, they're compact, they look kind of spherical, they're water soluble, and they're sensitive. If you change their environment, they're going to change their shape, and that's going to have a big impact on their function. Usually these globular functional groups or functional proteins will have functional regions or active sites. This is where they're going to interact with other molecules. So some examples might be antibodies. Antibodies will bind to antigens in your body. Hormones, which are going to bind to a receptor on a cell. Molecular chaperones that actually help other proteins fold. And the most important one that we're going to talk about now, or fairly soon, will be the enzymes. All right, so the function of these globular proteins depends on their shape, and they're sensitive to changes in their environment. Changes like pH or changing the temperature. So if one of these globular proteins does unfold or lose that shape, the active site becomes destroyed, it no longer works correctly. We call that denaturation. Its natural shape is what it began with. When you denature something, you change that natural shape. And it could be due to a variety of things. pH changes, changing the temperature, even just shaking it up too much could cause denaturation. Often this can be reversible, but sometimes irreversible. So a great example of protein denaturation is cooking an egg. All right, so I don't know how many of you guys cook, but if you take a raw egg out of the refrigerator and you break it, what color is the egg white? Clear, right? What happens to the color of that egg white if you put it over a hot stove? It becomes white. That's why we call it an egg white. So you have that egg white is a protein called albumin. Remember, see, it has the IN on the end. That means it's a protein. Now, this is a very important protein for the growth of an egg if it was fertilized and grew. It's also a really important protein that's found circulating in your blood. Right? So if you increase the temperature of it, it changes the shape of the albumin and it will go from clear to white. If you put that egg white now back in the refrigerator, will it ever change to clear again? No. You've permanently changed it. It's a irreversible denaturation. All right, so we're going to talk about a really important type of protein known as the enzymes. Enzymes are globular proteins that act as a biological catalyst. Now, a catalyst is anything that will speed up a chemical reaction, make a chemical reaction occur faster. So an enzyme can increase the speed of most of the chemical reactions that occur in your body. Let me correct that. Enzymes will control all the chemical reactions that occur in your body. Enzymes will operate mainly by lowering something we call the activation energy. And by doing that, increase the speed of a reaction. Activation energy is the energy needed to get a chemical reaction started. And all chemical reactions need an input of energy to make them occur. So if you can lower the amount of energy you need to get the chemical reaction to occur, you will increase how fast the reaction will occur. And these enzymes, different enzymes, will govern millions of reactions per minute in your body. So here's just an energy diagram of a chemical reaction. So down here at the bottom of this graph, let's work our way through this graph. We have time, or the progress of the reaction. And then on the side, we have the energy associated with the reaction. All right. So in any chemical reaction, we have reactants becoming products. Reactants yield products. Right? That's the general way we write most chemical reactions. So in this diagram, we have reactants. They're starting off at a higher energy, and they're becoming a product which has a lower energy. So in reality, we have a total amount of energy that is given off in this reaction. All right? But to make the reaction occur, we have to start off by adding a little bit of energy, and that takes time. That energy can come from many places. Could be some ATP. You've heard of ATP probably before, the energy currency of the cell. It might just be some heat energy from the body added in to allow the reaction to occur. All right? But initially, without an enzyme, that's a lot of activation energy that you have to use up just to make this reaction occur. If you add an enzyme, you'll notice that you lower the activation energy. So you have to add less ATP energy or less heat energy to make the reaction occur. So the reactions will occur faster if you have an enzyme. So enzymes are examples of functional or globular proteins, and they have an active site. So that active site has a shape that is specific for a substrate. Now here we're changing some terminology. Before I said that reactants become products. If, however, this reaction is sped up by an enzyme, we would call the reactants the substrates. So substrates become products. And those enzymes are specific for substrate. They only fit one or two types of molecules or one group of molecules. So they will fit perfectly with one type of a molecule. Another thing about enzymes, usually when we see them, their name ends with "ase." All right? And they often are named for the reaction they catalyze. So hydrolases or oxidases. So let's look at an example of one of the mechanisms of this enzyme action. Here we have an enzyme with a couple of active sites, and these active sites will fit the substrate, in this case a couple of amino acids, just fine. So the substrates bind at the active sites, they form an enzyme substrate complex, and in the process of the reaction you have a rearrangement between the two molecules, between the two amino acids. It still requires a little bit of an input of energy, but the activation energy is much lower than it would have been without the enzyme. This is a dehydration synthesis, so water is going to be released. And we end up with our product. Now, once those two amino acids are bonded together, they don't fit that enzyme anymore, and so they'll pop off that enzyme, and you will recycle that enzyme. So this enzyme is now available to come back and bind to another substrate. So a really important action of many of the proteins is they will be enzymes. And enzymes control every single chemical reaction that occurs in the body. So now let's move on to our last large organic molecule. This includes the nucleic acids. Now, probably some of you guys have heard of some of the nucleic acids. Two examples include deoxyribonucleic acid, DNA, or ribonucleic acid, RNA. These are really large molecules in the body. This DNA and RNA will contain carbon, hydrogen, and oxygen, just like we've seen in others, nitrogen, and also phosphorus. Once again, these are polymers with individual subunits being a nucleotide. So we get lots of nucleotides strung together. Each nucleotide has three main portions: a nitrogen-containing base, a pentose sugar, and a phosphate group. All right, so let's just look briefly at our two different types of nucleic acids. We have first deoxyribonucleic acid, or DNA. Now, all of the monomers, all of the nucleotides for DNA look almost identical. The only thing that's different will be what type of nitrogen-containing bases. Remember, all nucleic acid nucleotides, the building blocks, have three portions: the nitrogen-containing bases, the sugar, and also the phosphate group. So in all of these, they will have a phosphate group. The pentose sugar, the sugar will be deoxyribose. The only thing that changes is what type of nitrogen base you have. There are four different types: adenines, guanines, cytosines, and thymines, and each of those have different structures. There's two major groups, purines and pyrimidines. One group has a single ring, one group has a double ring, and they always base pair with each other within a double-stranded deoxynucleic acid structure. We have base pairing rulings. Adenine always base pairs with thymine. Guanine always base pairs with cytosine. We'll see an example of that in a few minutes. So it is double-stranded. There are two strands of this nucleic acid. The pentose sugar is deoxyribose. What does this do? It provides the instruction for a cell. All right, it's found in the nucleus of the cell. It contains the information needed to make proteins. Proteins, many of them will be enzymes. Enzymes control every single chemical reaction in the cell. So essentially, the DNA contains the information needed to control every chemical reaction in the cell. And another time you need your DNA is you replicate it. That means make an exact copy of it before cell division, so that each new cell being formed has an exact copy of that genetic information. Here's an example of the structure of DNA. You can see that it is double-stranded, and within the strand we can see that complementary base pairing. Let's look up at the top with the detail. We see that each individual nucleotide has three different components: the phosphate group, the sugar, deoxyribose, and the nitrogen-containing base. The first one you see up here is adenine, and it's always going to be base paired with a nucleotide containing thymine. So we can see that the thymine also has a phosphate group, a sugar, which will be deoxyribose, and this time we have a thymine as the nitrogen-containing base. These two nucleotides are held together across the two strands by hydrogen bonds. So hydrogen bonds were one of our types of chemical bonds. Sometimes a hydrogen bond will be an intermolecular bond. And I said intermolecular bond. That means between two molecules. So two water molecules attracted together. But sometimes hydrogen bonds can be an intramolecular bond. That means it's within a single molecule. Here's a great example. DNA has two different nucleotide strands. The two strands are held together by hydrogen bonds. Right? So this is a bond within the molecule. The other major type of nucleic acid includes the ribonucleic acid with 4 bases as well, but this time, well, you'll notice it still contains adenine, we still have a guanine, we still have a cytosine, but now instead of thymine, we have a uracil. Another change is the type of pentose sugar. It's now a ribose. Most of the time, we find RNA as a single-stranded molecule, not a double-stranded. And there are 3 main types of RNA molecules. First, we have messenger RNA. Messenger RNA is directly manufactured using one section of the DNA as a template. We copy it according to the sequence of nucleic acids on DNA. This is called messenger RNA because it contains the genetic message that it's going to take out to the rest of the cell. Then we have transfer RNA. Transfer RNAs will transfer an amino acid over to the messenger RNA. And then finally we have ribosomal RNA. Ribosomal RNA provides a site where the messenger RNA and the transfer RNA with its amino acid can get together. Now let's move on to another type of nucleic acid that probably a lot of you guys have heard of before. It's called adenosine triphosphate, or ATP. Now, adenosine, adenine, that sounds familiar, right? This is a form of a nucleic acid, so we are going to still see a nitrogen-containing base. In this case, it will be adenine. We're still going to see a pentose sugar. The pentose sugar will be a ribose. And we're still going to see phosphate, or in this case, multiple phosphates. So this is really important. It's the energy currency of the cell. It captures the energy that would have been found in glucose and then allows it to directly power chemical reactions. It's immediately available, easily usable by all body cells. So the structure of ATP will contain, it's an RNA nucleotide, it's adenine, it's the ribose sugar, and then 2 more phosphate groups. So if we look at our general ATP molecule, we see our adenine bound to our ribose sugar, that's called adenosine. If you only had 1 phosphate group, we would call that adenosine monophosphate. You add in another phosphate group, it's adenosine diphosphate. Add in that 3rd phosphate group, it's adenosine triphosphate. Now, you'll notice that these chemical bonds may be drawn a little bit strange. It's a little wiggly line like that. This symbolizes a high energy bond. It takes a lot of energy to hold this chemical bond together, and you gain a lot of energy when it's broken. So that's how ATP can transfer around chemical energy. So, when ATP does transfer that energy to another molecule, it does that by transferring a phosphate group to that other molecule. So when the ATP gives its energy to another molecule, that terminal phosphate, that's just the last phosphate in the series, is that terminal phosphate is transferred to another molecule, and that energizes the other molecule because it takes on that high energy bond. Those molecules are primed for cellular work. Work is life in motion. So just some examples of some cellular work driven by ATP. And we're going to look at a lot of these. Transport work, moving material across the cell membrane, especially if you're trying to move something across the membrane and you're trying to push it up a concentration gradient. That's going to require a lot of energy. Mechanical work, changing the shape of a cell, moving the body around. Or chemical work, right? Used to power chemical reactions. Those are all examples of energy driven by ATP, and ATP is one of the nucleic acids. So we have looked at all of the organic macromolecules. We've looked at carbohydrates and lipids in one of the videos, and proteins and nucleic acids in this second video of the organic macromolecules, of the type of organic molecules.